Good morning. It is Tuesday, the 18th of May. Let me know if you're here. Okay, let's talk about what we're on about today. I don't see any questions, which is a little bit surprising. So everybody was comfortable with all of the genetics problem-solving questions from yesterday? You were okay determining whether something was codominance or incomplete dominance? Okay, all right. Most people ask about question number 5, so I feel like every class has asked about that previous to this, so let's do it together. There's no shame in it, by the way, guys. If you have a question about one of the homework questions, I want to take them up with you. I'm ready to take them up. I usually don't do it unless students are gonna ask for it specifically, though, because if everybody's comfortable with it, I'm just wasting everybody's time. But so I don't want to do that either. Okay, I'm gonna start with number 5, and then Bria will do the other question that you mentioned there. Oh, you guys can't see this. Oh, this thing's going real slow. Oh, there she goes. Okay. Let me just read the question again here. 1, 5. Thalassemia, right. Okay, so thalassemia is a illness of anemia. Anemia is insufficient oxygen transportation in the blood. And I just have to really quickly refamiliarize myself with that. I think it's when you get a malformation of your red blood cells. Is that correct? Yes. Your red blood cells don't take on the correct shape, and so they don't bind to oxygen the same. And the question is, really, are all of your red blood cells going to do that? So, like, first describing the illness is very helpful in terms of determining, is it codominant or incomplete dominant? So we've got healthy red blood cells. And they basically look like little inner tubes. And they carry oxygen normally. And then you have, this is all described in the question, not the normal shaped red blood cells, but they mention that there is a minor thalassemia. Oh, is there 2 L's? No, 2 S's though. Thalassemia and a major thalassemia. And the difference between these two is that in minor thalassemia, you get some misshapen red blood cells, but some normal red blood cells. Okay, it's sort of like, it's like a half and half. And in major thalassemia, you get all misshapen red blood cells that do not hold oxygen very well. Okay, so that's the difference between them. So the question is asking, I believe, which is it, codominance or incomplete dominance, right? Because it says only one gene is involved. So first of all, you know that this is Mendelian. It's telling you that it's Mendelian. And there's only 2 alleles involved, which means that there's only 2 versions of the gene. So it's got to be codominance or incomplete dominance, because we have 3 phenotypes here. So it's something to note. There's 3 different phenotypes here. So 3 phenotypes means it's going to be incomplete or co-dominance. In regular dominance, incomplete dominance, there'd only be 2 phenotypes. So we know that for sure. What type of inheritance is it? And what would the genotypes be? Okay, so you need to basically we're deciding is it incomplete or codominance. So I feel like I'm always falling off the bottom of the screen. I gotta hunch over to get to my iPad. This is not really a great setup for this because I'm exploding my back. Hopefully you guys have a comfortable position wherever you're doing this. Okay, so remember the key here is remembering what the 2 different things mean, what incomplete dominance means and what codominance means. So incomplete dominance means that you can have 1 allele expressed or the other allele expressed, or the third allele, or not third allele, the third way to express it is the hybrid, right? It's when you have both different alleles. So let's say that let's write it as if it was that, okay? So let's say I'm going to use like B for my base letter. All right. So you could have healthy blood, or you could have the hybrid. I'll use T for thalassemic. Or you could have major thalassemia. Okay, let's say that that's the way that it broke down. So the question is, how is this middle one going to be expressed? Because we know that this one up here is going to be healthy, and this one down here is going to be thalassemic, like major thalassemic. So the middle one in incomplete dominance is a blend of the 2, which means every cell is going to be expressing both. Every cell is going to be expressing healthy and thalassemic at the same time. That's the pink, the idea of the pink plant, which is that it's making white. Every cell is making white and red pigment. So when you look at what actually is happening here in minor thalassemia, is every single cell expressing some thalassemic allele and some healthy? Or the alternative here would be so this would be I better actually write down what I'm saying here. So this is every cell expresses both, and you get a blend. The key here being that the middle one is every cell shows like a blend of the 2 traits together into a new third thing. Okay, or the other option is that it's codominance. And the actual way to write this out is going to be slightly different. So we still have capital letters for our base letters, but we just use small letters. So I can use the same letters, but this implies codominance here. And again, I know I've already mentioned this, but the textbook does not follow these guidelines for the notation, which is annoying. But I'm going to use them. Okay, so here we would have the obvious here. This is a healthy, right? And this is major thalassemia. But then the question is, what does the middle one look like? Well, in a codominant one, the middle one is each cell is either going to express one or the other. And so what you end up getting in the product at the end is both. Some cells do one and some cells do the other trait, but both traits are present. So the keyword word here being both. In codominant, both are shown. You can see both. And this is the idea of the some pink, some red hairs, some white hairs. The roan cow is codominant. Okay, I keep those two examples sort of in the back of my mind because that's like a comparison, a point of comparison always. Like, is it a pink example or is it a red and white example? So pink would be incomplete and red and white at the same time would be codominance. So which one are we looking at here? For minor thalassemia, the middle one, is it every cell expresses both and you get a blend? That'd be incomplete. Or is it some cells express one and some cells express the other, and so you can see both traits at the same time? Which one is it? Everyone fell asleep while I was doing this. That's okay. I won't hold it against you. I also realize there's a pretty long delay here, so. Right. You can see both at the same time. So which type of inheritance is it then, Logan? So incomplete would be if every cell has both healthy and thalassemic in it. In other words, all the cells look the same, Mila. Okay. No worries. So you got it here, Logan. Both traits are being expressed at the same time. Some in one cell and some in the other cell. And that's codominance. Okay, so this is an example of codominance. So ding ding ding, we have a winner. This is codominant inheritance pattern, and this is the notation that you would use. So to answer your question, Bree, yes, the textbook does not use the correct notation for codominance. Infuriating me. So I've talked to some other people about this. Most people that I met have learned it this way, that you use lowercase letters for superscripts for codominance to differentiate between the two of them. But not everybody. So not everybody I've met does that. Some of them treat codominance and incomplete dominance very similarly, and they have the same notation. So I don't know. I'm going to use this notation. I want to make it clear which type of inheritance pattern I'm talking about. But the textbook does not do that, which is very annoying. And I've spoken to some other biologists that say, well, yeah, not everybody does that. So fine, whatever. I'm going to do it. That's why I learned it. I can pull out my high school textbook. I have a copy of it right here. And it definitely does that. So maybe I'm just being old school. No, I don't think so. I'm pretty sure this is still done. So anyway, just that's to answer your question, Bree. So it says for number 2 on 196. I can actually read it, but yeah. So it says it's codominant, and then it shows you the notation for what should be incomplete dominance. The textbook doesn't follow the pattern that it's supposed to. I would just, me personally, when I wrote that down, I would do it the other way, but I'll leave it up to you. The textbook doesn't do it though. There's lots of other mistakes in the textbook that I've found over the years, so I'm gonna count it as a mistake. Any other questions from the homework? Hopefully that answered your question, by the way, about these two. That mixing up or having difficulty determining which it is, incomplete or codominance, is extremely common. That is, like, for students, a very, very common mix-up to make. It's why specifically why I include that question, like thinking through and deciding whether it's incomplete or co. And in order to answer it, you actually have to look up some information about thalassemia, because what they give you in the textbook is not enough information to decide whether it's incomplete or co. You could think that minor thalassemia is just every cell is a blended cell that is like slightly dysfunctional. That you could think that just from the description there. So that there's no way to actually answer that question without doing any research. I don't know what they're expecting in the text, but maybe they're assuming you're going to look it up. I did. Okay, I don't see any further questions. If there are, don't be afraid to put them in there, and as always, I will circle back. Let's talk about what we're up to today. Hopefully your exit quizzes went well. I know I've mentioned previously that you really should. You really should consider doing the exit quizzes, just to work out whether or not you have any little confusions that are sort of remaining in your understanding. Again, they don't count for marks, but I highly recommend them before the main quiz, and certainly you will see similar questions, so it'll be helpful. Okay, last major topic. We're going to talk about one more thing after this, but it's very small. We have one more type of inheritance to discuss. So we have been talking about complete dominance, the idea where there is a dominant and a recessive, and the dominant always shows if it's present, but if there's no dominant, then it's going to show the recessive. That's complete dominance. We've talked about the idea of incomplete and codominance. Okay, so both of them are ways that you can get sort of this hybrid gene expression when you have two alleles that are a hybrid. Basically, both alleles are present. Okay, and there is one last type of inheritance that we're going to talk about. There are more inheritance patterns, by the way, than just these. But these are sort of like, this is like your basic set. The other ones are just variations of these. The last one we're going to talk about is the idea of multiple allelic inheritance. So what happens with, instead of having just two alleles for something, what if there's 3 or 4 or 7? There technically can be infinite versions of a particular gene. So, well, maybe not infinite, but a very large number of possible configurations of a particular gene. So, obviously, we're not going to talk about those, but we are going to talk about one where there's 3. Okay, so one really common example of this, and it's the one that's used in every textbook because this is a real life example that is very common, is blood typing. There is more than one allele for blood type, and you guys probably have heard of this already. There is the A type, there is the B type, and there is the O type. Okay. There's also RH factor, rhodopsin factor, which is the positive, the plus or the minus, RH, or A positive, A negative, whatever. For the purposes of this example, we're going to completely ignore the rhodopsin factor. We're not even going to discuss RH positive and negative. So don't worry about that. I think it's mentioned in this video probably, but the negative or positive component, in terms of how that's determined genetically, it's on a separate gene, and we're just going to ignore it because that goes outside of the Mendelian inheritance that we're going to discuss in this course. So we're just going to talk about ABO, the ABO blood grouping. Okay, so how you get A, how you get B, and how you get O. Okay, so what I want you to do first is you're going to watch this video, which is just going to go over the basics of like, what does it mean for your blood to be A, and what does it mean to be O? And it's interesting, this actually comes back to something that we were talking about when we were talking about viruses, which is the idea of antigens. If you remember that antigens are just shapes. They are shapes on the surface of your cell. And the A's and the B's, that component of your blood is a shape. It is an antigen on the surface of your cell. It's A antigen and B antigen. And they'll talk more in detail in this video about them. But again, it's going back to the same idea here of shapes. Okay, and it also picks up on the immune system idea that we talked about earlier, which is that if you have the wrong shapes on something, your white blood cells will say, oh, I don't like that shape. We got to get rid of that. That's a bad shape in here. And it can do it to other human cells as well, for example, in cases of a blood transfusion. So anyway, I'm going to get you to watch this little video as an introduction. Okay, no worries, Mom. Thank you. And that'll give you sort of like just an intro into how blood typing works. I've pre-recorded the lesson for today. I did record this one quite a while ago, and so I make reference to timelines and things like this in this video that are obviously not relevant to this particular course because it was recorded at a different time. I actually recorded this last spring. This is my blood type video from last spring. It's one of my very first videos. Hopefully it's not terrible. But I do make reference to things like, oh, you know, like maybe you don't want to finish the assignments. Like, don't worry about it. We're not really marking those, or we're marking them, but it's not really going to hurt you or count against you or whatever on your mark. Anyway, I talk about that stuff. That's all stuff from last spring. You guys experienced that. You know what I'm talking about. But that doesn't obviously apply to this course. So just as a heads up, it's only at the beginning, I think, or the end I mentioned that. But just as a heads up, I've got emails before people saying, what? The assignments don't count? It's like, no, they count. That was last spring. Anyway, there are some questions that go along with this lesson from the textbook. It's more genetics problem solving, except this time it's with regards to blood typing and multiple allelic inheritance. Blood typing is, by the way, the only example of multiple allelic inheritance that we're going to discuss in this unit. It comes up one more time in the course in the evolution unit where we're going to talk about, where we're going to do a little lab activity, an online lab activity, where we look at insect color and the way that insects inherit their carapace color is multiple allelic. Anyway, we'll discuss it then at that point. But anyway, there's some practice problems 2 to 5 that are also in the course notes. I believe that I take those questions up at the end of this lesson, if I remember correctly. So it's like a pause and do them and then come back, which I mentioned in the lesson. If not, if you're having any questions about those practice problems on blood typing, please let me know, and I would be happy to go over them with you. And then there's a little exit quiz, as usual, at the end, okay? Just to test your knowledge on blood types and multiple allelic inheritance. That's our last sort of major topic of the unit. When you're done that, we are, you guys are going to do a little bit of reading. So what you're looking for is on page 14 of the genetics notes, there is a short little blurb on there about genetic illnesses. Let me pull it up for you so you can see it. And yes, there are the questions, by the way, that are in here. There are a number of things that we are skipping. So pedigrees, we talked about this just in passing. We're not going to cover it in the course anymore because we ran out of time. Sex-linked inheritance, really cool. That's when you get traits on your X and Y chromosome. It changes the inheritance pattern a little bit. No time. Skip it. And again, I mentioned there are other inheritance patterns, but again, they're just variations of the other ones. So the last thing that we are going to cover is this page right here on genetic disorders. So you are going to read this little blurb at the top here about genetic disorders, and then you're going to read in your textbook pages 204 to 207. Okay, this is also written on Brightspace. You can use that as an opportunity to add some last words here to your vocabulary list. You should be approaching 30 at this point, I would think. This unit has a tremendous amount of vocabulary, probably easily 10 to 15 words a day. So I would assume you probably have 30 already if you've been following along, but maybe not. If not, you'll find some new vocabulary here in 204 to 207. And then there are just some questions here that are about genetic disorders. They're also mentioned here on Brightspace, page 207, 1 to 5. Okay, I'm going to let you guys go off, do this work on your own. Basically, at that point, once you're done, this is the last day of the unit. That's right, guys. We made it to the end of genetics. So the rest of your time today, learning block 3, learning block 2, because I'm sure you'll finish this stuff during block 1. You probably will. Maybe you might just carry over into learning block 2 just a touch, but probably not much, depending on your reading speed, I suppose. But the rest of your time today is just to work on your meiosis live. So we talked about that previously, what you need to include. Consider watching the what's the word I'm looking for? Exemplar of some of the meiosis live presentation that I've put up there. It is a really good guide in terms of what you should include in your presentation. And then I'm here to support you. If you need any help with finding technology, looking for information, you're not sure about a particular part of the meiotic process, you're confused about where a certain number of chromosomes should be what it should look like, I can jump into a Google Meet with you anytime. I'm here to support you all day. So let me know if you need anything. You can type questions into the chat. You can also drop by during Learning Block 3 and chat. Remember that as we approach now the end of the unit, there are always 3 things coming due. It's always exactly the same at the end of every single unit. So you will have your quiz, which is available today, but you do not need to complete it until midnight tomorrow. Okay, midnight tomorrow would be like, that's when you should be done by then. If you need a little bit of extra time, it's no problem, but don't forget to email me and let me know that you need an extension. Okay? And remember always, if you're emailing me for extra time for stuff, just let me know what your timeline is. What is your proposed alternate deadline that you are going to stick to? Okay, it just makes the process easier, because then I don't have to respond and say, when do you feel you can complete this by? Because that's what I'm always going to ask you that. So just tell me when your proposed alternate deadline is. If it's reasonable, I'll say, cool, that's fine. So the idea here is always just to keep you on track and keep you being responsible for managing your own timelines. Okay, we should be doing that collectively, you and I together, to keep you on track. So anyway, quiz is up. You can complete it at any time. I recommend during Learning Block 3 today or tomorrow, so that you can ask questions about the quiz, but it's totally up to you when you want to do it. As always, there is a vocabulary list coming due, and it's always the day after at midnight, so tomorrow at midnight. But technically, at the end of today, you should be done, right? Because you've been adding words to it as we've been going, and so you just hand that in when you're done. And then you got your major assignment, the meiosis live, which hopefully you can be done by tomorrow midnight, but we'll see what happens. Anyway, you have time to work on it today. I realize that one takes a little bit longer, so if you need a little bit of extra time, not a huge deal, but please let me know. There is one additional thing at the end of this block, because tomorrow is your last day with me, and then you'll be heading off to your other course for your first course for another 2 weeks. So the last thing that you need is your homework questions, because I will have to be compiling your midterm marks after you go. I know it seems weird. It's weird timing. So I'll be giving you your midterm marks basically in the middle of your time with your other course, and you've only done 2 units. Whatever, we do what we can. So, but I do need your homework. So you'll notice here at the very bottom, there is a midterm homework questions portfolio. A number of people have asked me, where do I hand in my homework questions? So when you're done all the questions for this unit, you should have 2 documents. One of the homework questions from the first unit and one of the homework questions from the second unit. You're going to attach those to the homework questions portfolio and submit. Okay, and I use those to determine your learning skills. Are you completing work at home throughout the course? Okay, it's not meant to be an extra thing for you to do. It's just you just handing in the things you've already done. So you don't need to go back and do extra stuff. Just show me what you've done. Okay, and it's fine if you haven't done much. I mean, I shouldn't say it's fine. It's not great if you haven't done much, but I'm not expecting you to go back and do the homework. I'm just expecting you to attach what you have completed and submit. Okay, so there's technically 4 things, I guess, at the end of this unit. There's one extra thing, which is the homework questions portfolio. Okay, any questions about anything that's coming up here now? Timelines, what you're working on? Okay, I don't see any questions, so I'll let you guys get started. Please let me know if you need anything. Hey guys, welcome back for the second half. If you wouldn't mind letting me know if you are here. for the afternoon. Well, actually, I guess it's just morning part 2, whatever you want to call it. Give people a second to type if they're typing something. If not, by the way, there isn't any more. I would assume people are still probably working on the reading or the questions for the genetic illnesses portion. If people have any questions from there, you can feel free to ask too. Once you're done that, and done the genetic illness questions, you are complete the content for genetics. We are done genetics. You're welcome to write the quiz, although if you don't feel prepared right away, that's totally understandable. You want to take some time to review your notes, you're welcome to write it tomorrow. Learning block 3 is probably the ideal time. Okay, I'm not seeing any questions, so I will be here to support. If you need anything while you're working on your meiosis live, again, you're looking for some technology. You're looking for, like maybe some software or something to help with a particular objective that you have in your presentation, or you want to run something by me. Should I have 4 chromatids in this step? I'm having difficulty finding out where spermatogenesis happens in my species. Whatever question you happen to have, I'm here to help support you and help you find information. Even if now you're still a little bit hazy on how the process of meiosis works, and you want to go over that again, I would strongly urge you to drop by during Learning Block 3 so I could go through that with you in detail in the Google Meet. And I'm absolutely happy to do that. Okay, doesn't look like there's anything, so I will see you in the Google Meet or tomorrow morning, 8:20. Good luck, guys.